You are drifting above a living structure visible from space. Below, more than 2,600 kilometres of limestone stretch along the Australian coast, bright with colour, humming with life. In 1984, researchers on the Great Barrier Reef confirmed that over 100 coral species spawn on a single moonlit night. The builders have no brain, no bones, no eyes. Each one is smaller than a grain of rice, yet across millennia they have laid down billions of tons of stone. For decades, no one could explain how. Then came three converging discoveries: a symbiosis, a chemistry, and a celestial trigger. Together, they explain one of the most extraordinary construction projects in the history of life on Earth. Tonight, we descend to the reef, watch a polyp lay its skeleton, and witness the largest spawning event on the planet. If you enjoy what you hear, a like or subscribe goes a long way. The ocean is patient, so are we. From orbit, the east coast of Australia carries a scar of light. It runs for more than 2,600 kilometres along the continental shelf, from the warm shallows north of Cape York to the cooler waters off Bundaberg. And it is so vast that astronauts have described it as one of the few biological features on Earth recognisable from space without magnification. This is the Great Barrier Reef. Not a single reef, but a sprawling labyrinth of nearly 3,000 individual reef structures, threaded between more than 900 islands, draped across an area of ocean floor larger than the entire United Kingdom. It is the largest living construction on the planet, and nothing else comes close. You could lay the entirety of Italy across it, and still have reef left over at the edges. You could fly above it for 6 hours at the speed of a light aircraft, and never leave its borders. The numbers do not quite land the way they should, because the mind is not built to hold a living thing at this scale. 344,000 square kilometres of limestone, alive at the surface, dead and compacted below, and growing still. Think about that for a moment. The structure beneath the water is not a geological accident. It was not carved by rivers or thrust upward by tectonic forces. It was built, deliberately, chemically, relentlessly. And the builders are still at work. What makes this so difficult to accept is the nature of the builders themselves. They are not large, they are not strong. They are not, by any conventional measure, intelligent. The organisms responsible for the single greatest construction project in the history of life on Earth are soft-bodied animals smaller than a grain of rice, with no brain, no bones, no circulatory system, and no capacity for planning. Each one lives and dies within a space you could cover with a fingernail. Each one contributes a quantity of limestone so small it would be invisible to the naked eye. And yet, multiplied across billions of individuals and thousands of years, their accumulated effort has produced a structure weighing billions of tons, visible from beyond the atmosphere. This is not an engineering feat in the way we usually understand the term. There is no blueprint, no foreman, no communication between workers about the larger shape of what they are creating. Every individual animal simply does what its biology compels it to do, and the reef is what emerges. The magnificent is not designed. It is inherited, compounded, stacked one invisible layer at a time across millennia, until the result becomes something no single organism could ever have imagined, had it the capacity to imagine anything at all. Aboriginal and Torres Strait Islander peoples have known this reef for over 60,000 years. Their sea country traditions carry knowledge of its rhythms, its creatures, and its changes across deep time, passed from generation to generation long before Western science arrived with its instruments and its categories. The reef existed before agriculture, before writing, before cities. The living surface you see today is between 6,000 and 8,000 years old, but the limestone platform beneath it stretches back hundreds of thousands of years, built and rebuilt through cycles of growth and collapse that dwarf the entire span of human civilization. And it all begins with a single animal, no larger than the head of a pin, doing something extraordinary with seawater. Imagine an animal the size of a pencil tip, soft, translucent, vaguely cylindrical, open at one end like a tiny cup. Around the rim of that cup, a ring of delicate tentacles waves slowly in the current, each one armed with microscopic stinging cells capable of paralysing prey far smaller than itself. At the base of the cup, a simple digestive cavity. No heart, no lungs, no blood, no skeleton of its own, at least not one it was born with. This is a coral polyp, and it belongs to the phylum Cnidaria, the same ancient lineage that produced jellyfish and sea anemones. If you held one in your hand, you would barely feel it. If you dropped it on a table, it would be almost invisible. Most coral polyps measure between 1 and 3 millimeters across. Some species produce polyps as small as a fraction of a millimeter. Others, the so-called mushroom corals, can reach several centimeters, but these are the exception. The vast majority are tiny, soft, and unremarkable to look at. They feed at night, extending their tentacles to capture drifting particles of organic matter, microscopic organisms, and the occasional larva unlucky enough to drift within reach. During the day, most species retract into their stony homes and wait. But here is the detail that changes everything. The coral polyp you are picturing, this fragile, translucent animal barely visible to the naked eye, is doing something that almost no other soft-bodied creature on Earth can do. It is building a house out of rock, not metaphorically, not approximately. The polyp is extracting calcium ions and carbonate ions from the seawater around it, combining them within a specialised layer of tissue on its underside, and precipitating them into solid, crystalline calcium carbonate. The mineral it produces is called aragonite, a specific form of limestone, and the polyp deposits it in a thin, continuous layer beneath and around its own body, constructing a rigid protective cup called a corallite. The animal lives inside this cup. It is both the architect and the sole resident. The tissue responsible for this work is called the calicoblastic epithelium, a layer of specialised cells pressed directly against the growing skeleton surface. Between those cells and the skeleton itself lies a vanishingly thin space filled with fluid, the extracellular calcifying medium, and it is within this confined space that the chemistry of reef building actually happens. The polyp actively pumps hydrogen ions out of this fluid, raising its pH, making it more alkaline, and in doing so it shifts the chemistry in favour of calcium carbonate precipitation. The fluid becomes supersaturated. Crystals begin to form. The skeleton grows. This is not a passive process. The coral is not simply waiting for minerals to settle out of the water the way salt crystallizes at the bottom of an evaporating dish. It is actively driving the chemistry, spending energy to manipulate the conditions inside that tiny pocket of fluid so that stone forms where stone would not otherwise form. Every fraction of a millimetre of skeleton represents a deliberate metabolic investment. The animal is manufacturing rock, and the rock it manufactures is not random. The aragonite crystals grow in radiating bundles called spherulites, fanning outward from dense nucleation points, known as centres of calcification. A team working out of the University of Wisconsin-Madison demonstrated that this spherulitic architecture is not merely decorative. It is the fastest way to fill three-dimensional space with aragonite crystals. The geometry is optimised. Aragonite grows roughly 10 times faster, along one crystal axis than along the others, and the spherulite arrangement exploits this anisotropy, allowing the skeleton to fill space more rapidly and more uniformly than any other crystal growth pattern could achieve. The result is a structure that is both mechanically strong and isotropic, meaning it resists force equally from all direction. It is, in a real sense, a form of biological three-dimensional printing, and the coral has been doing it for over 200 million years. Here is what makes this even more remarkable. A single polyp working alone, building its tiny cup of aragonite grain by grain, would produce nothing of consequence. A speck of limestone on the ocean floor, invisible, irrelevant. But coral polyps do not work alone. They reproduce asexually, budding off exact genetic copies of themselves, and those copies remain physically attached to the parent, sharing tissue, sharing nutrients, forming a connected living sheet that spreads across the growing skeleton. One polyp becomes two, two become 10, 10 become 1,000, 1,000 become 1,000,000, and every single one of them is building simultaneously on the same expanding platform of stone. When a polyp dies, its soft tissue decays, but the limestone cup it built remains, permanently. The next generation settles on top of it and begins building again, adding their own layer of stone to the layer beneath. Generation after generation, century after century, the structure grows. The living coral on a reef is only a thin veneer on the surface. A few millimeters of active tissue stretched over what may be meters, tens of meters, or even hundreds of meters of dead skeleton below. You're looking at a graveyard that is also a construction site, a monument built from the accumulated remains of every generation that came before, each one adding its invisible contribution to a structure that none of them will ever see completed. What comes next is the secret that makes all of this possible, because the polyp alone cannot build a reef. It does not have the energy. The chemistry of calcification is expensive, and a tiny animal feeding on whatever drifts past its tentacles at night simply cannot fuel the kind of relentless, continuous construction that reef building demands. Something else is needed. Something inside the coral itself, hidden in its own tissue, performing a feat of chemistry that transforms everything. If you were to peer inside the tissue of almost any reef-building coral, you would find them. Millions of them, packed into every square centimetre of the animal's living surface, nestled within the cells of the inner tissue layer like tiny stowaways. They are single-celled algae, members of the family Symbiodaniaceae, and they are so abundant that they give the coral most of its visible colour. The greens, the golds, the chocolate browns of a healthy reef are not the colours of the animals themselves. They are the colours of the algae living inside them. These organisms are photosynthetic. They capture sunlight, combine it with carbon dioxide and water, and produce sugars and oxygen exactly as a plant does. And here is the arrangement that changes everything. Up to 90% of the sugar they produce is transferred directly to the coral host. The polyp does not earn this energy by hunting. It does not chase it, capture it, or compete for it. It receives it continuously as long as the sun is shining from billions of microscopic power stations embedded in its own flesh. This is the engine. Without it, the polyp is a tiny predator, scraping by on whatever drift into its tentacles at night. With it, the polyp is a construction machine, flushed with energy, capable of driving the expensive chemistry of calcification hour after hour, day after day, year after year. The symbiosis does not merely help the coral build, it makes reef building possible. But the chemistry it fuels is far more sophisticated than anyone suspected for most of the 20th century, and the story of how we came to understand it is one of the most quietly dramatic revolutions in marine science. For decades, the prevailing view of coral calcification was essentially physical. The idea was straightforward. Seawater is naturally supersaturated with calcium carbonate in warm, shallow tropical waters, and the coral simply provides a surface on which crystals can nucleate and grow. The animal creates the conditions, the physics does the rest. Under this model, the polyp is less an architect than a scaffold. It holds a position in the water column, and the minerals precipitate more or less on their own. This was a reasonable assumption. Aragonite does form spontaneously in supersaturated solutions. But it was wrong, or at least profoundly incomplete, and the evidence against it accumulated slowly from laboratories scattered across the world until the weight of it could no longer be ignored. The first cracks appeared when researchers began measuring the chemistry inside the calcifying space itself, that vanishingly thin pocket of fluid between the calicoblastic cells and the skeleton surface. What they found did not match the surrounding seawater. The pH was higher, sometimes by a full unit. The concentration of dissolved inorganic carbon was elevated. The aragonite saturation state, the measure of how favourable the conditions are for crystal formation, was far above anything the ocean could produce on its own. The coral was not passively allowing precipitation. It was actively engineering the chemical environment inside its own body to force it. Then came the discovery that changed the framework entirely. A team working at Rutgers University used an approach no one had tried before on coral skeletons. They combined ultra-high resolution three-dimensional imaging with solid-state nuclear magnetic resonance spectroscopy, and what they found overturned the physical model for good. The mineral deposition was not beginning as ordered crystals nucleating from a supersaturated solution. It was beginning as something far stranger. Amorphous calcium carbonate, a disordered, unstable form of the mineral with no crystal structure at all, was being deposited first in tiny nanoparticles within microenvironments rich in organic molecules secreted by the coral itself. Only after these amorphous particles were in place did they begin to reorganize, aggregate, and crystallize into the ordered aragonite fibers that make up the mature skeleton. Think about what this means. The coral is not waiting for crystals to grow. It is manufacturing a precursor material, placing it precisely where it needs to go, and then allowing it to transform into the final product. The organic molecules it secretes, a cocktail of proteins rich in acidic amino acids, appear to stabilize the amorphous phase, prevent premature crystallization, and guide the eventual formation of ordered aragonite at specific nucleation sites. The skeleton is not precipitated, it is assembled. The distinction matters enormously, because it means the coral has far more control over its own construction than anyone previously believed. This carries a profound implication. If the skeleton was simply a product of seawater chemistry, then any change in ocean conditions, a drop in pH, a reduction in carbonate ion concentration would directly and proportionally reduce the coral's ability to build. But if the coral is biologically controlling the process, manipulating its internal chemistry independently of the surrounding water, then it may have a degree of resilience that the purely physical model could never predict. The animal is not at the mercy of the ocean. It is negotiating with it. Whether that negotiation has limits is another question entirely, and one that the science has not yet fully answered. There is growing evidence that as ocean pH continues to fall, even the coral's internal buffering capacity begins to falter. The skeleton still forms, but it forms thinner, less dense, more porous. The house is still being built, but the walls are getting weaker. The calcifying fluid can only be pushed so far above the chemistry of the water surrounding it. At some point, the energy cost of maintaining that internal advantage becomes unsustainable. That point may be closer than we would like, but under the conditions that have prevailed for most of the last several thousand years, the system works beautifully. The polyp draws calcium from the seawater, pumps hydrogen ions out of the calcifying space to raise the pH, secretes organic matrix molecules to guide crystal nucleation, deposits amorphous precursor nanoparticles, and then allows those particles to mature into crystalline aragonite in radiating spherolytic bundles that fill space with extraordinary efficiency. All of this happens in a fluid-filled pocket thinner than a human hair, powered by sugar from symbiotic algae, repeated billions of times across the surface of a single reef. The chemistry is elegant, the scale is staggering, and the structures it produces are not uniform. They are wildly, extravagantly diverse, because the way a colony grows depends on species, on genetics, on light, on current, on the shape of the substrate beneath it, and on the competitive pressures of everything growing around it. The result is a reef that looks less like a wall and more like an underwater city, with a skyline shaped by hundreds of different architects working simultaneously in different styles. Consider the range. Massive corals, the brain corals and boulder corals of the genus Porites, grow in dense, rounded heads that can reach several meters in diameter and live for centuries. Their growth is slow, sometimes less than a centimetre per year, but their skeletons are extraordinarily dense and resilient, and a single colony the size of a small car may be 400 or 500 years old. These are the anchors of the reef, the heavy foundations that resist storm damage and provide stability to everything around them. At the other extreme, branching corals like the staghorn acropora grow with reckless speed, pushing slender fingers and antler-like branches outward and upwards at rates of 10 to 15 centimetres per year. They are the fast colonisers, the opportunists, the species that race to fill open space after a disturbance. Their skeletons are lighter, more porous, and far more fragile, but their growth rate means they can dominate a reef within years of a cyclone clearing the slate. A thicket of staghorn coral provides an intricate labyrinth of shelter. Thousands of narrow passages between branches where juvenile fish hide, shrimp forage, and tiny crabs establish territories barely larger than a coin. Between these extremes lies every imaginable variation. Plating corals extend broad, flat shelves outwards from the reef face, maximising the surface area exposed to light for their symbiotic algae. Encrusting species spread in thin sheets across rock and rubble, binding loose fragments into solid reef framework. Foliose corals grow in ruffled, leaf-like folds. Columnar species rise in pillars. Fan corals, though technically not stony corals at all, add their own flexible, branching architecture to the three-dimensional complexity of the reef face. Every form fills a different ecological role, and every form creates a different kind of space for other organisms to inhabit. What unites all of these growth forms is the underlying biology. Every colony, regardless of its shape, is a connected network of genetically identical polyps, joined by a continuous layer of living tissue called the choanosarc. This tissue stretches across the entire surface of the skeleton, linking polyp to polyp, allowing nutrients, signalling molecules, and energy to be shared across the colony. A coral head the size of a table is not a collection of independent animals. It is a single organism, a superorganism if you prefer, thousands or millions of polyps operating as one coordinated living system. injure one part of the colony and the surrounding tissue responds. Shade one section, and the energy produced by sunlit polyps on the other side is redistributed to compensate. This connected architecture is what allows the colony to behave as something greater than the sum of its polyps. It can allocate resources. It can mount immune responses. It can shift energy toward growth at the margins when space opens up, or toward repair when part of the skeleton is damaged. Modern reef science increasingly treats the coral not as a single animal, but as a holobiont, a functional unit comprising the polyp, its symbiotic algae, a community of associated bacteria, viruses, archaea, and fungi, all working in concert. The colony is not just a builder, it is an ecosystem within an ecosystem, and the reef it constructs is an ecosystem within that. And the construction never stops. When a polyp dies, its soft tissue disappears within days, consumed by bacteria and scavengers. But the limestone cup it built endures. It becomes substrate, it becomes foundation. The next generation of polyps, whether budded from the same colony or settled from the open water as planular larvae, builds directly on top of it. Layer upon layer, generation upon generation, the skeleton grows upward and outward. The living surface of the reef is a veneer, a skin of active tissue perhaps a few millimetres thick, stretched over a monument of dead stone that may extend tens of metres down to the original substrate. Everything you see when you look at a reef, the colour, the movement, the life, exists in that thin upper film. Everything below it is history, compressed into rock, silent and permanent. What emerges from this process, given enough time and enough favourable conditions, is not just a colony, it is a platform, a geological feature, a mountain of biogenic limestone rising from the seafloor, built entirely by animals that individually weigh less than a raindrop. And the question that hangs over all of it, the question that determines whether this platform becomes a reef or remains a scattered collection of isolated colonies, is energy. Where does the energy come from to sustain construction on this scale, in waters so nutrient poor that they are sometimes called blue deserts? The answer has been hiding in plain sight, in the colour of the coral itself, and it is one of the most consequential partnerships in the history of life on Earth. The partnership is ancient. Molecular clock analyses suggest it began roughly 160 million years ago, deep in the Jurassic period, when the ancestors of modern reef-building corals first took in photosynthetic algae and discovered that the arrangement was worth keeping. Dinosaurs still roamed the continents. The Atlantic Ocean was barely a crack between the land masses that would become Africa and South America. And somewhere in a shallow tropical sea, a coral and an alga struck a deal that neither has broken since. The algae belong to the family Symbiodiniaceae, a diverse group of single-celled dinoflagellates once classified under the single genus Symbiodinium before modern genetics revealed just how varied they really are. They are spherical, golden brown, and so small that roughly 1 million of them can occupy a single square centimetre of coral tissue. They live inside the cells of the coral's inner tissue layer, the gastrodermis, each one enclosed within a membrane-bound compartment provided by the host. They are not passengers. They are residents, embedded so deeply in the animal's biology that removing them is like pulling the engine from a car. The vehicle still exists. It simply cannot go anywhere. What these algae do is photosynthesize. They absorb sunlight, capture carbon dioxide, and produce energy-rich organic compounds, sugars, glycerol, amino acids, the molecular fuel that powers all cellular work. In a free-living alga, those compounds would be consumed by the organism that made them. but inside a coral, the arrangement is different. The host has evolved mechanisms to intercept the algae's photosynthetic output, and the transfer is not modest. Up to 90% of the carbon fixed by the zooxanthellae is handed over to the coral. The animal receives a continuous stream of high-quality energy without lifting a tentacle. In return, the coral provides the algae with a stable home, a protected environment bathed in light, and a steady supply of the raw materials photosynthesis requires. Carbon dioxide, produced as a waste product of the coral's own respiration, is delivered directly to the algae. Nitrogen and phosphorus, scarce in the oligotrophic tropical waters where reefs grow, are recycled from the coral's metabolic waste. The arrangement is efficient to the point of elegance. Almost nothing is lost. The waste of one partner becomes the fuel of the other, and the system runs in a tight, self-sustaining loop that has kept reefs growing in some of the most nutrient-poor waters on Earth for millions of years. This is the reason coral reefs exist where they do. Tropical oceans are famously unproductive. The warm surface waters are stratified, cut off from the nutrient-rich deep water below, and the result is vast stretches of clear blue sea with very little life. Blue deserts, marine biologists sometimes call them. And yet it is precisely here, in these barren waters, that the most biodiverse ecosystems in the ocean are found. The paradox dissolves the moment you understand the symbiosis. The corals do not need the ocean to be productive. They carry their own productivity inside them. The energy the zooxanthellae provide does more than feed the polyp. It drives the construction. Two researchers working in Jamaica in the late 1950s made the observation that would anchor an entire field of study. Thomas Garreau and Nora Garreau measured the rate at which corals deposited calcium carbonate under different light conditions and found something striking. In the light, calcification rates surged. In the dark, they dropped dramatically. The pattern was consistent, repeatable, and unmistakable. Light was not merely helpful for coral growth, it was essential. They called the phenomenon light-enhanced calcification, and it became one of the foundational principles of reef science. The mechanism behind it is still debated in its finer details, but the broad picture is clear. Photosynthesis by the zooxanthellae removes carbon dioxide from the coral's internal environment. This shifts the carbonate chemistry in a direction that favours the precipitation of calcium carbonate. At the same time, the sugars produced by the algae provide the metabolic energy the polyp needs to actively pump ions across membranes, maintain the elevated pH in the calcifying space, and synthesize the organic matrix molecules that guide crystal formation. The algae are not directly building the skeleton, but they are providing both the chemical conditions and the energy supply that make construction possible at reef building rates. Without them, calcification slows to a fraction of its normal pace. The polyp can still deposit some limestone, feeding on whatever it catches at night, but nowhere near enough to outpace the constant erosion, dissolution, and wave damage that attack the reef from every direction. This is why reef-building corals are confined to the photic zone, the uppermost layer of the ocean where sunlight penetrates in sufficient intensity to drive photosynthesis. Most reef growth occurs in the top 10 to 20 metres of water, where light is strongest. Below that, the energy subsidy from the algae diminishes, calcification slows, and the competitive advantage that allows corals to dominate shallow tropical waters gradually disappears. By 100 metres depth, the light is too dim for most zooxanthellate corals to survive. The reef is, in the most literal sense, solar-powered. Its geographic limits, its depth limits, and its rate of growth are all set by the availability of sunlight to a symbiotic alga living inside the tissue of an animal that builds with stone. The symbiosis is productive, ancient, and remarkably successful, but it has a flaw, a catastrophic one, and it is written into the very chemistry that makes the partnership work. The photosynthetic machinery inside the zooxanthellae is extraordinarily sensitive to temperature, not to cold particularly, but to heat. When the water surrounding a coral warms beyond a narrow threshold, typically just 1 to 2 degrees Celsius above the normal summer maximum for that location, the algae's photosynthetic apparatus begins to malfunction. The protein complexes in the thylakoid membranes that capture light energy and pass it down the electron transport chain start to degrade. The system that normally converts sunlight into chemical energy with elegant precision begins to misfire, and when it does, it produces something dangerous. Instead of passing electrons neatly from one carrier to the next, the damaged photosystem begins generating reactive oxygen species, highly unstable molecules that attack and destroy cellular structures indiscriminately. Membranes are shredded. Proteins are denatured. The damage cascades. Here is the cruel irony. The algae that have been feeding the coral, powering its construction, sustaining the entire reef, become toxic. The very partnership that made the reef possible now threatens to destroy it, and the coral responds in the only way it can. It expels the algae. Millions of zooxanthellae are ejected from the coral's tissues, cast out into the surrounding water, and the polyp is left alone, stripped of its energy supply, its colour, and its capacity for sustained calcification. This is coral bleaching. The name comes from what it looks like. Without the golden brown pigments of the zooxanthellae, the coral's translucent tissue reveals the white limestone skeleton beneath. A bleached reef does not look dead exactly, but it looks emptied, drained. The colour is gone. The warmth is gone. What remains is a ghostly white landscape that seems to belong to a different world. A bleached coral is not immediately dead. The polyp is still alive, still capable of feeding on whatever particles drift past its tentacles. If temperatures drop within a matter of weeks, the coral can reacquire zooxanthellae from the surrounding water, repopulate its tissues, and recover. Some corals have demonstrated a capacity to take up different strains of algae after bleaching, strains that may be more tolerant of elevated temperatures than the ones they lost. This flexibility offers a thread of hope, but it is a thin one, because recovery depends entirely on the stress being temporary. If the elevated temperatures persist for too long, the coral starves. Without the energy subsidy from its algae, it cannot maintain its tissues, cannot repair damage, cannot fight off disease, and it dies. The skeleton remains, but the living veneer is gone, and what was a vibrant, growing structure becomes inert rock, quickly colonized by filamentous algae and boring organisms that begin to dismantle it from within. And here is where the paradox deepens. Research has shown that in many cases, the expelled zooxanthellae are still photosynthetically healthy. Their light harvesting machinery is functioning. They can still produce sugar. The breakdown is not a failure of the algae to photosynthesize. It is a failure of the transfer. Under thermal stress, the normal flow of photosynthetic products from alga to host is disrupted. The algae begin to retain resources for their own survival. The relationship shifts. What was mutualism tips toward parasitism? The coral, sensing that it is paying the metabolic cost of housing an organism that is no longer sharing its output, ejects it. The bleaching is not a malfunction. It is a decision made at the cellular level to cut losses and survive alone rather than maintain a partnership that has turned toxic. The frequency of these events is accelerating. In the 1980s, mass bleaching was rare, documented only a handful of times. By the 2000s, it was occurring every few years. A researcher at James Cook University compiled the most comprehensive record ever assembled of mass coral bleaching across the globe, and found that the average interval between severe events had halved. Where reefs once had a decade or more to recover between bleaching episodes, they now face repeated thermal stress with only a few years of reprieve. The Great Barrier Reef alone experienced severe mass bleaching in 2016, 2017, 2020, 2022, and 2024. 5 events in 9 years, each one weakening the reef further, each one reducing the window for recovery. The temperature threshold is not a distant theoretical danger. It has already been crossed, repeatedly, and the crossings are becoming more frequent as global ocean temperatures continue to rise. A temperature increase of roughly 0.7 degrees Celsius per century may sound modest, but for an organism whose symbiotic partnership collapses at just 1 to 2 degrees above its normal summer peak, that rate of change is not gradual. It is existential. And yet the reef is still here. Damaged, diminished, stressed beyond anything it has experienced in its recent geological past, but still here, still building, still laying down limestone, grain by grain, polyp by polyp, powered by an algal partnership that has survived 160 million years of upheaval, and is now facing a challenge unlike any it has encountered before. Whether it can adapt fast enough, whether the corals that harbour heat-tolerant algal strains can outcompete those that do not, whether the pace of warming will outstrip the pace of biological adjustment, these remain open questions. The answers are not yet written. What is certain is that the reef has faced collapse before and returned. Understanding how it did so, how fast it built, how it recovered, and what set the limits of its resilience requires something the symbiosis alone cannot provide. It requires time. And the story of how corals use time, how they convert centuries into meters of stone, is where the construction truly reveals its scale. 0.5 centimetre. That is what a typical massive coral adds to its skeleton in a year. 0.5 centimetre of new aragonite, deposited grain by grain across the entire living surface of the colony, laid down through the sustained chemical effort of every polyp working in concert with its symbiotic algae. In a good year, with warm, stable water, plentiful light, and no significant stress events, that figure might reach 2 centimetres. Under truly ideal conditions, certain species have been recorded growing at 4 to 5 centimetres annually, but these are the exceptions. For the great boulder corals and brain corals that form the structural backbone of a reef, the pace is measured in millimetres. Sit with that number for a moment, 0.5 centimetre per year. A brain coral the size of a football has been growing for several decades. A massive porites colony the size of a car, the kind you might swim past on a reef and barely glance at, could be 300 or 400 years old. It was alive before the Industrial Revolution, before the French Revolution. It was building its skeleton while Mozart was composing, while empires were rising and collapsing on the other side of the world, and it was doing the same thing then that it is doing now, adding 0.5 centimetre of limestone to its surface, year after year, with a patience that makes human timekeeping feel absurd. These growth rates are not guesses. They are readable directly from the skeleton itself, because corals lay down their limestone in annual bands, much as trees lay down rings of wood. A dense band deposited during the warm, high-growth summer alternates with a less dense band from the cooler winter months, and the result is a record of growth encoded permanently in the stone. Cut a coral core from a massive colony, polish the cross-section, and you can count the years the way a forester counts rings. The science of reading these bands is called sclerochronology, and it has turned coral skeletons into some of the most valuable environmental archives on Earth. The chemistry of each band records the temperature of the water in which it was deposited, the salinity, even the trace chemical signatures of distant volcanic eruptions and changes in rainfall patterns on the nearest landmass. A single coral head can hold centuries of climate data in its bones. But growth rate is only half the story. A reef does not simply grow upward, it is simultaneously being torn apart. From the moment a coral deposits its first grain of limestone, the forces of destruction are already at work. Parrotfish are among the most conspicuous agents. They graze the reef surface constantly, scraping algae from the limestone with fused beak-like teeth strong enough to bite through solid rock. Each bite removes a sliver of skeleton along with the algae. A single large parrotfish can consume and excrete more than 300 kilograms of limestone in a year, grinding it through a specialised pharyngeal mill and depositing it as fine white carbonate sand. The white beaches of tropical islands, the ones you see in photographs, the ones that look like they were poured from a bag of powdered sugar, are substantially made from this process. That sand passed through a fish. Beneath the surface, the destruction is quieter but no less relentless. Boring sponges of the genus Cleona tunnel through the reef framework, dissolving limestone chemically and excavating it physically, creating a labyrinth of internal cavities that weaken the structure from within. Lithophagous bivalves, clams that bore into rock, carve cylindrical holes. Polychaete worms thread their way through the matrix. Sea urchins rasp the outer surface with rows of tiny teeth. Together these organisms are called bioeroders, and they are not anomalies or pests. They are an essential part of the reef's economy, recycling solid limestone back into loose sediment, freeing up space, and creating the fine carbonate rubble and sand that fills gaps, settles into crevices, and eventually cements into new reef rock. The health of a reef is defined by the balance between these two forces. Construction on one side, destruction on the other. The net result, the actual rate at which the reef accumulates new limestone after subtracting everything lost to bioerosion, wave damage, storm breakage, and chemical dissolution, is called the reef accretion rate. On a healthy reef, accretion is positive. The builders are winning, but only just. The margin is thin. A reef that grows 2 centimetres of new skeleton per year, but loses 1.5 centimetres to erosion, is accreting at only half a centimetre annually. That slender surplus, compounded over millennia, is what produces the massive limestone platforms we see today. But if anything tips the balance, if growth slows due to bleaching or acidification while bioerosion continues at its usual pace, the equation reverses. The reef begins to dissolve. Not dramatically, not in a single catastrophic event, but steadily, imperceptibly, the structure shrinking rather than growing, losing height, losing complexity, losing the three-dimensional architecture that everything else depends on. There is one more builder that deserves mention, because without it, the reef would be a pile of rubble rather than a solid wall. Crustow's coralline algae, the pink and purple encrustations that coat rock surfaces in the highest energy zones of the reef, serve as the reef's mortar. These calcifying algae spread in thin, hard sheets over exposed limestone, binding loose fragments together, cementing broken coral branches into solid substrate, and reinforcing the reef crest where wave energy is greatest. They are not glamorous, they are easily overlooked, but structurally, they are indispensable. A reef without crustose coralline algae would be a loose aggregate of coral skeletons, vulnerable to every storm, incapable of maintaining the steep vertical faces and solid ramparts that define a mature reef front. Together, the corals, the coralline algae, the foraminifera, the mollusks, and the echinoderms that contribute their own calcium carbonate shells and spines to the matrix, create something that transcends biology. They create geology, and the reef they construct behaves not as a living community, but as a landform, a feature of the Earth's surface with mass, permanence, and a physical presence that outlasts any individual organism by orders of magnitude. The living coral on a reef is a veneer. This is perhaps the most important single fact about reef construction, and it is the one most easily missed. When you swim over a coral reef and see the vivid colours, the swaying tentacles, the fish darting between branches, you're seeing only the outermost skin of a structure that extends far, far below. That living layer is a few millimetres thick. Beneath it, sometimes directly beneath, sometimes separated by thin layers of sediment and encrustation, lies the accumulated skeleton of every coral generation that came before. Metres of it. Tens of metres. On some reefs, hundreds of metres, stretching down through the water column and into the continental shelf itself. The Great Barrier Reef's modern living surface sits atop a limestone platform that in places exceeds 100 metres in thickness. Drill into it and you pass through layer after layer of fossil coral, cemented sediment, and compacted rubble, each stratum representing a period of growth, followed by a period of death, followed by regrowth, followed by more death. The oldest material at the base of some reef structures dates back hundreds of thousands of years. The living tissue on top is adding its own contribution right now, this year, this month, and it will become part of that record in turn. The reef does not distinguish between past and present, it simply accumulates. This accumulation is what creates the physical complexity that makes reefs so extraordinary as habitats. A flat sheet of coral growing on a flat seafloor would produce nothing more than a limestone pavement. But reefs do not grow flat. They grow upward at different rates in different places, depending on species composition, light availability, water flow, and the geometry of the underlying substrate. Faster growing colonies overtop slower ones. Sections of reef collapse under their own weight or break during storms, creating rubble slopes and overhangs. Bioerosion carves caves and tunnels. Sediment fills low points. The result, over centuries and millennia, is a structure of extraordinary three-dimensional complexity. Vertical walls dropping into deep water, shallow sandy lagoons, narrow channels where currents accelerate, cavernous overhangs where light never reaches, rubble fields colonised by encrusting organisms, and towering coral bommies rising from the sea floor like underwater skyscrapers. Every one of these features is a habitat. Every cave shelters a different community. Every crevice hosts a different assemblage of invertebrates. The structural complexity of the reef is not incidental to its biodiversity. It is the cause of it. Without the three-dimensional architecture that millennia of coral growth have produced, the reef would support a fraction of the species it currently sustains. The animals did not choose to build a complex structure. They built what their biology dictated, and the complexity emerged from the accumulation of countless simple acts of construction and destruction playing out over timescales that no single organism could witness. And the reef is not unique in this. The geological record is littered with ancient reef structures built by organisms long extinct, visible today not as underwater marvels, but as exposed rock formations on dry land. The Capitan Reef of West Texas, now the dramatic cliffs of Guadalupe Mountains National Park, was a thriving reef in the Permian period, roughly 260 million years ago. The Dolomite of northern Italy, those jagged alpine peaks beloved by mountaineers and tourists, are built substantially from Triassic reef limestone, the fossilised remains of organisms that built in shallow tropical seas when that part of Europe lay near the equator. These ancient reefs are reminders that what the Great Barrier Reef is doing today is not new. It is one of the oldest construction traditions on Earth, carried forward by successive lineages of organisms across hundreds of millions of years of evolutionary time. The current builders, the scleractinian corals, have held the contract for roughly 240 million years. They inherited it from the rugose and tabulate corals that built the great reefs of the Paleozoic, and they have been refining their technique ever since. The materials are the same, the chemistry is the same, the pace is the same. What changes is the world around them, the temperature of the water, the chemistry of the ocean, the level of the sea, and how effectively the corals can keep building through whatever the planet throws at them. How they reproduce, how they spread to new locations, how they seed the growth of entirely new reefs across hundreds of kilometres of open ocean, is the next part of the story. And it begins with moonlight. Once a year, on a handful of nights in late spring, the Great Barrier Reef does something that no one believed was possible until it was witnessed firsthand. It reproduces all at once. The process begins in silence, in the hours after sunset, when the water above the reef is warm and still and dark. Then, from the surface of coral colonies scattered across hundreds of kilometres of reef, tiny bundles begin to rise. Each bundle is a sphere no larger than a pinhead, a packet of eggs and sperm wrapped together in a lipid coating, buoyant enough to drift upward through the water column toward the surface. One bundle appears, then another, then a dozen, 100, 1,000, until the water above the reef is thick with them. A slow-motion blizzard of pink, red, orange and white rising through the darkness like confetti falling in reverse. Divers who have witnessed it describe the sensation as otherworldly. The beams of their torches catch millions of tiny spheres drifting upward simultaneously, the entire reef exhaling its future into the sea. This is mass coral spawning, and it is one of the largest synchronized reproductive events in the natural world. On the Great Barrier Reef, more than 130 species of coral participate. They release their gametes within the same narrow window, often within the same hour, on the same few nights each year. The timing is not approximate. It is precise to a degree that seems impossible for organisms with no central nervous system, no calendar, and no means of communicating with one another across the vast distances of the reef. For most of the 20th century, coral reproduction was assumed to be a modest, localized affair. The prevailing view held that most coral species were brooders, fertilizing their eggs internally and releasing fully developed larvae that settled nearby. Broadcast spawning, the explosive release of eggs and sperm into open water, was considered uncommon. The reef was thought to reproduce the way a garden grows, quietly, incrementally, each colony handling its own affairs in isolation. That assumption was shattered in the early 1980s. A group of researchers working at 3 locations on the Great Barrier Reef spent 2 consecutive spring seasons observing coral reproduction at night, and what they documented was so unexpected that it took the broader scientific community years to fully absorb. Across all 3 sites, over a total of 32 species, Coral colonies release their gamete bundles within the same few nights following the late spring full moon. The spawning was synchronous across species, across sites, and across reef structures separated by considerable distance. It was not a quiet, localised process. It was an event, a coordinated mass release on a scale that no one had imagined. The paper describing these observations landed in 1984, and it did more than document a new phenomenon. It overturned a fundamental assumption about how reef-building corals reproduce. External fertilisation and broadcast spawning were not exceptions, they were the rule. Within 2 years, a follow-up study expanded the list to 105 scleractinian species observed spawning synchronously on the same reef system. The scale was almost incomprehensible. Over 100 species, spanning multiple families, releasing their reproductive material into the water at almost exactly the same time, on the same nights, year after year. The question that followed was obvious. How? These are not organisms with sophisticated sensory systems. A coral polyp has no eyes, no brain, no hormonal communication network linking it to distant colonies of different species on distant reefs. And yet they achieve a level of reproductive synchrony that would challenge any coordinated system designed by engineers. Something in the environment must be cueing them, and whatever it is must be precise enough to synchronise spawning not just within a colony, not just within a species, but across the entire reef community. The answer, it turned out, operates on 3 nested timescales, each governed by a different environmental signal. The broadest cue is temperature. Coral spawning on the Great Barrier Reef occurs in late spring during the period of most rapid seasonal warming. Research examining spawning times across global reef sites found that it is not the absolute temperature that matters, but the rate of temperature increase. Corals spawn during the month when the sea surface temperatures are rising most steeply. This sets the calendar. It narrows the window from 12 months to 1. Within that month, the specific nights are determined by the lunar cycle. Spawning occurs reliably within a few days of the full moon, most commonly between the 3rd and 6th nights after it. This has been confirmed across reefs in the Pacific, the Indian Ocean, the Caribbean, and the Red Sea. The full moon acts as a synchronizing signal, a clock visible across the entire reef simultaneously resetting the reproductive countdown each month until the temperature conditions are right. And within those nights, the precise hour is set by the fading of daylight. Spawning typically begins between 1 and 3 hours after sunset, with different species staggering their release in a sequence that is remarkably consistent from year to year. One species may begin releasing at 7:30, another at 8, another at 9:30. The sequence is predictable enough that experienced observers can tell what time it is by which species is currently spawning. Think about the elegance of this system. Temperature selects the month. Moonlight selects the night. Sunset selects the hour. 3 environmental signals, layered on top of one another, converging to synchronize the reproductive effort of over 100 species across thousands of square kilometres of reef. No communication is required, no coordination between colonies. Each polyp simply reads the same environmental cues and responds on the same schedule. The synchrony is emergent, arising not from any central control, but from the fact that every organism on the reef is listening to the same sky. The staggering of species within a single night serves its own purpose. By releasing gametes at slightly different times, closely related species reduce the risk of hybridization. The eggs of one species are already dispersing by the time the sperm of a similar species enters the water. The timing is tight enough to ensure fertilization within species, where the concentration of compatible gametes is highest in the mynates immediately after release, but offset enough to minimize cross-contamination between species. It is a choreography performed in darkness, without rehearsal, by organisms that have been repeating it for millions of years. But how does a coral polyp, an animal without eyes, detect moonlight? How does it distinguish between a waxing gibbous and a full moon? How does a creature with no brain track the passage of days since the last lunar peak? These are not rhetorical questions. They are among the most extraordinary problems in sensory biology, and the answers have only begun to emerge in the last decade. A team based in Israel set out to identify the molecular basis of lunar detection in the staghorn coral Acropora millipora, one of the dominant spawning species on the Great Barrier Reef. They tracked gene expression across entire lunar cycles and found that changes in moonlight intensity triggered cascades of molecular signalling within the coral tissue. Specific photoreceptor proteins, closely related to melanopsin, the light-sensing molecule found in the non-visual photoreceptors of vertebrate eyes, were identified in the coral. These receptors do not form images. They do not allow the coral to see, but they are exquisitely sensitive to changes in the intensity and spectrum of ambient light, and they respond to the waxing and waning of the moon with measurable shifts in gene expression, neuropeptide production, and downstream hormonal signalling. The coral cannot see the moon, but it can feel it chemically in the changing light levels filtering through the water column night after night. A different group, working in Taiwan, approached the same question from the opposite direction and arrived at a complementary answer. Studying the coral Dipsastria speciosa, they discovered that moonlight does not trigger spawning. It suppresses it. As long as bright moonlight is present in the sky during the early hours of the night, the reproductive cascade is held in check. Spawning occurs not when the moon is present, but when it is temporarily absent, during the dark interval between sunset and moonrise that occurs on the nights following the full moon. Here is why this matters. After a full moon, the moon rises progressively later each evening. On the first night after the full moon, it appears shortly after sunset, and the sky is bright all night. By the third or fourth night, there is a gap, a window of complete darkness between sunset and moonrise, and it is during this window that spawning peaks. The darkness is the trigger. The removal of the suppressive moonlight signal releases the reproductive cascade, and the gamete bundles begin to rise. The two mechanisms are not contradictory. They describe different aspects of the same system, and they may operate in different species, or even in different populations of the same species. What unites them is the principle. Coral reproductive timing is governed by light, specifically by the predictable, cyclical changes in nocturnal illumination produced by the lunar orbit. The coral is using the moon as a clock, not by reading the time, but by responding to a rhythmic environmental signal that has been cycling with absolute regularity for as long as there have been oceans on Earth. The sophistication of this is difficult to overstate. These are organisms without a centralized nervous system. They have no circadian clock in the way mammals do, no suprachiasmatic nucleus coordinating daily rhythms, and yet they maintain reproductive timing synchronized to celestial events with a precision measured in hours. The biology required to achieve this, the photoreceptors, the signaling cascades, the molecular timekeeping mechanisms, represents a form of biological chronometry that has been refined by natural selection across tens of millions of generations. And it is vulnerable. Research conducted over 3 decades on Red Sea coral communities revealed something alarming. several species that had historically spawned in tight synchrony were losing their coordination. Spawning events that once involved entire communities releasing gametes on the same nights were becoming fragmented, with different colonies of the same species spawning weeks or even months apart. The reproductive synchrony was breaking down. 2 researchers at Tel Aviv University documented this shift and identified it as a previously unrecognized threat to reef persistence. If corals cannot spawn together, fertilization rates collapse. Eggs released into water without compatible sperm are wasted. Sperm released when no eggs are present disperses and dies. The entire reproductive strategy depends on concentration, on overwhelming the water with gametes simultaneously so that the probability of any single egg encountering a compatible sperm cell is high enough to sustain the population. When synchrony breaks down, recruitment fails. No new larvae settle. The existing colonies age without replacement. The reef does not die in a visible dramatic event. It simply stops reproducing, and the decline that follows is slow, silent, and almost invisible until the population is too depleted to recover. The causes are not fully understood, but the leading candidates are precisely the environmental changes already stressing reefs from every other direction. Rising water temperatures may be shifting thermal cues that set the spawning month. Artificial light from coastal development may be interfering with the moonlight signals that set the night. Degraded water quality may be altering the chemical environment in ways that disrupt hormonal signalling. Each of these individually might be manageable. Together, they represent a threat to the one process that allows reefs to replenish themselves. A reef that cannot spawn is a reef that cannot persist. It does not matter how fast the existing corals grow, how resistant they are to bleaching, or how well they compete for space. Without sexual reproduction, without the annual explosion of genetic diversity that mass spawning represents, the reef has no future beyond the lifespan of its current inhabitants. The moonlit trigger is not a curiosity. It is the mechanism on which everything depends. And what happens after the gametes reach the surface, after fertilization occurs in the warm water of a spring night, after the tiny embryo begins its journey as a drifting speck in an ocean of impossible vastness, is a story of chance, survival, and the founding of new worlds. At the surface, the gamete bundles break apart. Eggs and sperm disperse into the warm water, and a fertilization occurs within minutes. The meeting is chemical, guided by species-specific signaling molecules that help sperm locate compatible eggs amid the vast soup of reproductive material from dozens of species released on the same night. For a brief window, the upper water column is dense with possibility, then the moment passes. Unfertilized eggs decay, excess sperm dies, and the fertilized embryos, the tiny fraction that found their match, begin to develop. Within hours, the embryo has divided enough times to form a hollow ball of cells. Within a day or two, it has elongated into a plannular larva, a creature barely visible to the naked eye, pear-shaped, covered in fine hair-like cilia that beat in coordinated waves to propel it slowly through the water. It has no mouth, no skeleton, no tentacles, no means of feeding. It survives entirely on the energy reserves packed into the original egg by its parent colony. It is, in the most literal sense, running on inherited fuel, and the clock is ticking. The plannula drifts. It is carried by surface currents, by tidal flows, by the slow circulation patterns of the tropical ocean. Some larvae travel only meters from the reef where they were spawned. Others are swept into open water and carried for days, sometimes weeks, covering distances of tens or even hundreds of kilometres, before they either find a suitable place to settle or exhaust their energy reserves and die. The range of dispersal is enormous, and it is largely beyond the lava's control. It can swim weakly, adjusting its position in the water column, moving toward or away from light, rising or sinking in response to chemical gradients. But against the force of ocean currents, these adjustments are marginal. The planula is a passenger, and the ocean is choosing its destination. The odds against any individual larva are extraordinary. The vast majority will never reach adulthood. They are eaten by fish, by filter-feeding invertebrates, by the myriad organisms that exploit the annual bonanza of spawning as a food source. They are swept into unsuitable waters, too deep, too cold, too far from any reef. They settle on surfaces that cannot support them. Sand that shifts, rock that is already claimed, dead coral smothered in algae. They run out of energy before they find a home. Of the millions of planulae produced by a single colony in a single spawning event, perhaps a handful will survive to establish new colonies. Perhaps none. The lottery is brutal and the house always wins. But the lottery only needs to pay out occasionally. A single successful settlement, one planula finding the right surface at the right time, can found a colony that persists for centuries and produces millions of its own larvae in return. The mathematics of reproduction at this scale are built on staggering waste. The system does not need efficiency. It needs volume. And volume is exactly what mass spawning provides. When a planula does find a suitable surface, the transformation is swift. The larva descends from the water column, contacts the substrate, and begins to test it. This is not random. The planula is reading chemical signals from the surface beneath it, and the cues it responds to are remarkably specific. Surfaces encrusted with crustose, coralline algae are strongly preferred. These calcifying algae release chemical compounds, including specific peptides and polysaccharides that act as settlement inducers. Molecular signals that tell the drifting larva, this is a reef, this is where corals grow, this is a good place to begin. The communication is ancient, spanning an evolutionary gulf of hundreds of millions of years between the alga producing the signal and the coral larva responding to it. Two organisms that have never met, that belong to entirely different kingdoms of life, coordinating through chemistry alone. If the cues are right, the planula attaches. It flattens against the substrate, cements itself in place, and begins to metamorphose. The ciliated, free-swimming larva transforms into a sessile polyp, developing a mouth, tentacles, and the beginnings of a digestive cavity. Within days, it begins to secrete its first calcium carbonate, the initial thin disc of aragonite that will become the foundation of an entire colony. The founding polyp has landed. The reef, in that precise spot, has begun. From this single polyp, through asexual budding, a colony will grow. It may take years for the colony to reach a size visible to a passing diver. It may take decades for it to become a significant structural element of the reef. But the process is underway, and every massive coral head, every branching thicket, every encrusting sheet on every reef on Earth traces its origin back to a moment exactly like this one. A drifting speck of tissue carried by currents it could not control, landing on a surface it did not choose, and building. Research conducted on the central Great Barrier Reef revealed something important about the spatial pattern of this process. Recruitment was measured at increasing distances from an isolated platform reef, and the results showed that coral larvae do not disperse evenly. The highest concentrations of new recruits were found close to the parent reef, in zones of high water residence time where currents circled back and retained larvae in the local area. Recruitment dropped sharply with distance, falling by an order of the magnitude within little more than a kilometre. The implication was striking. Reefs are largely self-seeding. The majority of larvae that successfully settle on a reef were spawned by that same reef. The vast plumes of gametes released during mass spawning may drift spectacular distances, but the colonists that matter most, the ones that actually survive and build, tend to stay close to home. This does not mean long-distance dispersal is irrelevant. Over evolutionary timescales, even rare events of successful distant settlements are enough to maintain genetic connectivity between reef systems separated by hundreds of kilometres of open ocean. A single larva arriving from a distant reef and surviving to adulthood introduces new genetic material into the local population. And over thousands of generations, this trickle of gene flow prevents isolated reef communities from diverging into separate species. The mass spawning is both a local replenishment strategy and a long-range genetic insurance policy operating simultaneously on two entirely different timescales. And when you step back far enough, when you stop thinking about individual polyps and individual reefs and begin to consider what happens when these organisms build continuously across geological time, the picture changes dramatically. Because a reef is not a fixed object. It is a living system that responds to its environment. And the environment it responds to most powerfully is the level of the sea. A young volcanic island rising from the tropical ocean provides the first foothold. Its rocky shores, washed by warm, clear water, are ideal substrate for coral settlement. Planulae arrive, attach, metamorphose, and begin to build. The colonies expand, merge, and eventually form a continuous band of reef hugging the coastline, growing outward from the shore into shallow water. This is a fringing reef, the simplest and most direct form of reef construction. A living border of limestone pressed against the land. A naturalist aboard a survey vessel in the 1830s saw these fringing reefs on islands across the Pacific and the Indian Ocean, and he noticed something. On some islands, the reef was not pressed against the shore at all. It stood some distance offshore, separated from the land by a deep lagoon of calm, clear water. These were barrier reefs, structurally similar to fringing reefs, but displaced outward, with a navigable channel of water between the reef crest and the island's coast. On still other islands, the island itself was gone entirely, and all that remained was a ring of reef enclosing a shallow central lagoon with no land at all. These were atolls, and they were among the most mysterious landforms in the ocean. That naturalist was aboard the Beagle, and the theory he constructed to explain these three reef types was among the most elegant geological arguments of the 19th century. He had spent years on the west coast of South America observing the evidence of crustal uplift, land rising slowly out of the sea, carrying with it the fossilized shells of marine organisms found thousands of feet above sea level. If land could rise, he reasoned, it could also sink. And if a volcanic island surrounded by a fringing reef were to subside slowly into the ocean, the reef, provided its upward growth could keep pace with the sinking of its foundation, would gradually separate from the receding shoreline. The lagoon between reef and island would widen. The barrier reef stage would begin. And if the island continued to sink until it disappeared entirely beneath the waves, only the reef would remain, a ring of coral marking the former outline of a vanished island. An atoll. The theory was published in 1842 as the first monograph produced from the voyage, years before the work on natural selection that would later make its author the most famous scientist in history. It was conceived before he had ever set foot on a coral island, worked out from first principles while the Beagle crossed the Pacific, and completed in draft by November of 1835. The survey of the Keeling Islands in the Indian Ocean, where the Beagle's crew spent days charting the atoll's lagoon and reef structures, provided confirming observations. The theory was simple, beautiful, and testable, and when it was presented to the Geological Society of London, even Charles Lyell, whose own volcanic crater hypothesis it directly contradicted, conceded that the younger man's explanation was superior. But confirmation on the ground, or rather beneath it, proved maddeningly difficult to obtain. If the theory was correct, then beneath any atoll there should be an enormous thickness of coral limestone, built up over the millennia as the reef grew upward to keep pace with the subsiding island beneath it. Drilling into an atoll to test this prediction was attempted in the 1890s on the island of Funafuti, reaching over 300 metres of depth and still finding coral, but the results were considered inconclusive at the time. The definitive test came in the 1950s, when the United States military, preparing to conduct nuclear weapons tests at Enuetik Atoll in the Marshall Islands, drilled exploratory boreholes through the reef. The drill passed through over 1,200 metres of coral limestone, before finally striking the volcanic basalt of the original island beneath. The theory was vindicated. The reef had been growing upward for millions of years, tracking the slow descent of its volcanic foundation into the Pacific. The picture, however, has proven more complicated than the original model allowed. A marine geologist who spent more than 40 years studying reef systems pointed out a fundamental gap in the reasoning. The 1842 theory was conceived in an era before the discovery of ice ages. The concept that global sea levels could rise and fall dramatically, driven by the growth and retreat of massive continental ice sheets, was unknown, and it turns out that these cyclic changes in sea level are at least as important as subsidence in shaping atolls. During glacial periods, when vast quantities of water were locked in ice, and the sea levels dropped by over 100 metres, the tops of carbonate platforms were exposed to air, and rainwater slowly dissolved the limestone, carving bowl-shaped depressions in the centre. When the ice melted and sea levels rose again, coral recolonised the now hollowed platforms, growing most vigorously around the raised rims where conditions were best, producing the ring-shaped structure characteristic of an atoll. The shape is not solely a product of upward growth on a sinking island. It is also a product of dissolution during periods of exposure, sculpted by rain and then reclaimed by coral when the water returned. The map that accompanied the 1842 monograph, a hand-compiled chart marking the location of every known reef on Earth, has been compared to modern satellite imagery and found to be remarkably accurate. The classification of reefs into fringing, barrier, and atoll types remains the standard framework used by marine geologists today. The core insight that reef morphology is shaped by the interplay between coral growth and changes in the relative position of land and sea remains correct. What has changed is the understanding of what drives those changes. Not just subsidence, not just sea level rise, but a complex interplay of tectonic movements, glacial cycles, dissolution, and biological response that has been playing out across millions of years. And the record of that interplay, written in the limestone of the reef itself, is what reveals just how resilient these structures have been, and what it took to break them. The Great Barrier Reef has died before. Not once. 5 times. This is not speculation. It is written in the rock. In 2018, the results of a 10-year multinational research effort were published, and they told a story that rewrote the biography of the world's largest reef system. A team led out of the University of Sydney, working with collaborators from Spain, Japan, Belgium, France, the United States, the United Kingdom and Australia, had drilled into the submerged fossil reefs along the outer edge of the Great Barrier Reef's continental shelf and extracted cores of ancient limestone stretching back 30,000 years. The cores contained layer upon layer of fossil coral, cemented sediment, and biological debris. And when they were dated and analysed, they revealed not a single continuous history of growth, but a sequence of catastrophic collapses followed, each time by remarkable recovery. The drilling was conducted as part of a major ocean research expedition, and the sites were chosen deliberately, 16 locations along the shelf edge near Cairns and Mackay, where the reef had built and rebuilt itself through some of the most dramatic environmental upheaval in recent geological history. The period covered by the cores spans the approach to the last glacial maximum, the Ice Age itself, the subsequent deglaciation, and the establishment of the modern reef. It is a window into how the reef responded to changes in sea level, ocean temperature, and water quality that make anything in recorded human history look trivial by comparison. The first two death events occurred as the Ice Age deepened. Around 30,000 years ago, and again around 22,000 years ago, falling sea levels exposed the reef to air. The corals, which can only survive submerged, were killed by the very thing they had been building toward, their own proximity to the surface. As the ocean retreated, the reef crest broke through into the atmosphere and died. But the reef did not simply accept its fate. It migrated. As the water pulled back, new coral growth established itself further down the continental slope, tracking the receding shoreline, colonising deeper substrate, and rebuilding at a lower elevation. The reef moved seaward, chasing the water. Then the ice began to melt. Sea levels rose, rapidly at times, and the reef was forced to reverse direction. Two more death events followed, around 17,000 and 13,000 years ago, this time caused not by exposure, but by drowning. The water rose so fast that the corals could not grow upward quickly enough to stay within the photic zone. They were submerged beneath water too deep for their symbiotic algae to receive sufficient light, and they died. Once again, the reef responded by migrating, this time landwards, establishing new growth on the flooding continental shelf as it became available. The fifth death event, around 10,000 years ago, appears different from the others. The evidence in the cores points not to sea level change as the primary driver, but to a massive influx of sediment. Terrestrial material washed from the land as rising seas reshaped the coastline, smothered the reef, blocking light, clogging polyps, and burying the delicate living surface beneath layers of mud and silt. The reef that had survived glaciation and deglaciation, that had tracked sea levels across more than 100 metres of vertical range, was brought down by dirt. Think about the resilience this record reveals. Over 30,000 years, the Great Barrier Reef experienced 5 separate events severe enough to cause widespread death across the system. Each time it recovered. Each time it rebuilt. Sometimes by migrating laterally across the seafloor, sometimes by growing vertically to track rising water, sometimes by recolonising entirely new substrate. The speed of its recovery was not uniform. Some rebuilding took centuries, some took millennia, but the pattern is unmistakable. The reef is not a fragile ornament, it is a survivor, a system with a deep capacity for regeneration built into its biology and its reproductive strategy. Not every reef system survived these transitions. The record also contains what geologists call give-up reefs, structures that could not keep pace with the rate of environmental change and were permanently drowned, buried, or abandoned. These are the reefs that lost the race, the ones whose growth rate was outstripped by the speed of rising water, or whose substrate was rendered uninhabitable by sediment. They are visible today as submerged terraces on the outer shelf, fossil limestone platforms that mark the positions where the reef once stood before the ocean overtook it. The distinction between a keep-up reef, one that successfully tracked environmental change and survived, and a give-up reef, one that did not, often came down to remarkably fine margins. A few millimetres per year of additional growth, a slightly lower rate of sea level rise, a marginally cleaner water column. The difference between survival and extinction was never dramatic. It was incremental, decided by the same slow arithmetic of accretion that governed everything else about reef construction. The modern living reef, the one visible from space, the one that supports one of the most diverse ecosystems on the planet, is the latest iteration of this cycle. It established itself on the flooded continental shelf roughly 6,000 to 8,000 years ago. As post-glacial sea levels stabilised near their present height and warm, shallow water spread across the broad platform that is now the Queensland coast. The reef we see today is young by geological standards, younger than the pyramids of Egypt might suggest, but the limestone platform beneath it carries the memory of every previous incarnation. Layer upon layer, death upon death, growth upon growth, the entire 30,000-year history compressed into stone beneath a living skin no thicker than a fingernail. The research team's lead author expressed grave concern about whether the reef's proven resilience would be sufficient to survive the current combination of stresses. Past sea surface temperature changes occurred over timescales of thousands of years. Current warming is happening over decades. Past sediment influxes were driven by natural coastal processes. Current sediment loads are amplified by agriculture, land clearing, and coastal development along the Queensland coast. The reef has faced each of these challenges individually and survived. Whether it can survive them simultaneously, at the pace they are now arriving, is the question the geological record cannot answer. What it can answer is what the reef does when it is healthy, when the conditions are right, when the water is warm and clear, and the corals are growing as they were designed to grow. And what it does is build a world. A mature coral reef is not a surface, it is a volume. Centuries of growth, collapse, regrowth, bioerosion, and sedimentation produce a three-dimensional structure of almost incomprehensible complexity. Vertical walls drop from the reef crest into deep water, their faces encrusted with fans, whips, and sponges. Overhangs extend outward like stone awnings, casting permanent shadows where light-sensitive organisms cluster. Caves penetrate deep into the reef body, their interiors cool and dark, inhabited by species that never see sunlight. Narrow channels between reef formations funnel currents, creating rivers of fast-moving water that carry plankton and nutrients past the waiting tentacles of filter feeders. Rubble fields at the base of the reef slope provide a different kind of habitat entirely, a shifting, unstable landscape colonised by organisms adapted to disturbance. Sandy lagoons behind the reef crest offer calm, shallow water where seagrass beds and juvenile fish nurseries flourish. Every one of these features exists, or because corals built it, died, and left their skeletons behind for the next generation to modify. The architecture was not planned. It emerged from the accumulated consequences of biological construction and physical destruction operating over timescales far longer than any individual organism could witness. And it is this architecture, This three-dimensional structural complexity that explains the single most astonishing statistic in marine ecology. Coral reefs cover less than 1% of the ocean floor. Less than 1%. And yet they support roughly a quarter of all known marine species. The disproportion is staggering. An area of ocean smaller than the state of Texas harbours a diversity of life that rivals anything found in the remaining 99% of the marine environment combined. The Great Barrier Reef alone is home to more than 1,600 species of fish, over 600 species of coral, 133 species of shark and ray, 30 species of whale and dolphin, and 6 of the world's 7 species of marine turtle. The numbers are not the point. The point is the density, the sheer concentration of different forms of life crammed into a habitat built entirely by other forms of life. The explanation lies in structure. A flat, featureless seafloor offers a limited range of conditions. A coral reef offers thousands. Every cave is a different temperature. Every crevice is a different light level. Every channel is a different current speed. Every overhang provides a different degree of shelter. The organisms that inhabit the reef do not simply live on it. They partition it, dividing the three-dimensional space into microhabitats so fine-grained that hundreds of species can coexist within a single square metre of reef face without directly competing. A cleaner wrasse establishes a station on an exposed coral head, servicing client fish that queue for parasite removal. A moray eel occupies a hole in the reef wall 2 metres away, emerging only at night to hunt. A family of clownfish defends an anemone on the reef crest, never straying more than a body length from its stinging tentacles. A hawksbill turtle rests on a ledge at 15 metres depth, returning to the same spot day after day. Each organism occupies a niche defined not by geography but by the microarchitecture of the reef itself, and the diversity runs deeper than the eye can see. Among the most abundant vertebrates on a coral reef are fish so small and so well hidden that they were largely overlooked until recently. Cryptobenthic fish, species typically less than 5 centimetres long, live in the interstices of the reef, in the tiny cracks and gaps and hollows that riddle the limestone surface. They are gobies, blennies, cardinalfish and triple fins, and they are extraordinarily numerous. Research has shown that they form a critical base of the reef food web, reproducing rapidly, dying young, and being consumed in vast quantities by the larger fish that draw human attention. They are the unseen engine of the reef's trophic economy, converting the energy stored in algae and detritus into fish biomass that cascades upward through the food chain. The reef has a soundscape too. A healthy reef is not silent. It hums, crackles, and pops with the combined noise of snapping shrimp, grinding parrotfish, drumming damselfish, and the collective feeding sounds of thousands of organisms going about their business. This sound carries through the water, and it has a purpose beyond ambience. Research has demonstrated that the larvae of reef fish and the planulae of corals can detect the low-frequency sound signature of a healthy reef from considerable distances in open water and use it as a navigational beacon, swimming toward the noise, orienting their settlement toward the source of the sound. A degraded reef, one that has lost its fish and its invertebrate community, grows quieter, and the silence is not just an absence. It is a failure of communication, a missing signal that larvae in the open ocean can no longer hear, reducing the probability that new recruits will find their way to the reef at all. The reef builds itself from stone. It fills itself with life. It advertises its own existence through sound. And every element of this system, the structure, the diversity, the acoustics, the food web, depends on the continued growth of organisms that individually weigh almost nothing, live for years at most, and contribute a quantity of limestone so small it would be lost in a teaspoon of sand. The contrast between the scale of the builders and the scale of what they build remains, even now, one of the most quietly extraordinary facts in all of biology. But the reef is not only a cradle of life, it is also a factory, one that processes raw materials from the ocean on an industrial scale and deposits them as solid rock on the continental shelf. The scope of that factory and the forces that constrain it reveal the reef not as a local phenomenon, but as a geological power with global reach. Every reef is a quarry, not in the metaphorical sense, though the comparison holds, but in the most literal way imaginable. The reef is being mined, ground down, dissolved, and excavated every hour of every day by organisms that have made the destruction of limestone their entire way of life. And without them, the reef as we know it could not exist. The most visible demolition crew works in plain sight. Parrotfish patrol the reef surface in loose schools, their fused beak-like jaws biting directly into the limestone, scraping away the thin film of algae that grows on exposed skeleton and taking a layer of solid rock with every bite. The sound is audible underwater, a constant rhythmic crunching that forms part of the reef's background noise, as distinctive in its way as the snapping of shrimp or the popping of coral feeding. Each bite is small. Each fish takes thousands of bites a day. And the limestone they consume does not stay inside them. It passes through a specialised pharyngeal mill, a set of grinding plates in the throat that pulverise the calcium carbonate into fine particles, and it emerges at the other end as sand. White, fine-grained, carbonate sand. A single large humphead parrotfish can process more than 5 tons of reef material in a year, excreting well over 300 kilograms of it as clean white sediment. Scale that across the thousands of parrotfish grazing a single reef system, and the numbers become staggering. The white sand beaches of the Maldives, of Hawaii, of the Caribbean, of every tropical postcard destination on Earth, are built substantially from this material. You have almost certainly walked on it. You have almost certainly let it run through your fingers. It passed through a fish. But parrotfish are only the most conspicuous part of a much larger economy of destruction. Beneath the reef surface, a hidden workforce operates around the clock. Boring sponges of the genus Cleona are among the most destructive. They settle on exposed limestone, extend fine filament into the rock, and dissolve it chemically from within, excavating networks of tunnels and chambers that riddle the reef framework like Swiss cheese. A heavily infested coral head can lose a significant fraction of its structural integrity to sponge boring alone, the solid skeleton reduced to a fragile lattice that collapses under its own weight or shatters in the next storm. Lithophagus bivalves, clams that bore into rock using a combination of chemical secretions and mechanical abrasion, carve cylindrical holes that penetrate deep into the reef body. Polychaete worms thread through the matrix in sinuous tunnels. Sea urchins scrape the outer surface with rows of tiny calcified teeth, each pass removing a fine layer of limestone and leaving characteristic grazing scars behind. Together, these organisms are called bioeroders, and their collective effect is enormous. On some reefs, bioerosion removes as much calcium carbonate annually as the corals deposit. The reef is not a structure that simply grows. It is a structure caught in a permanent tug of war between the forces that build it and the forces that tear it down, and the outcome of that war determines whether the reef grows taller, stays the same height, or begins to shrink. Here is where the balance becomes precarious. On a healthy reef, construction narrowly outpaces destruction. The margin is often remarkably thin, a few millimetres of net accretion per year, but compounded over centuries and millennia. That thin margin is what produces the massive limestone platforms visible from space. When the builders falter, when bleaching kills the coral, or acidification slows its calcification rate, the bioeroders do not stop. They continue at the same pace, indifferent to the condition of the living surface above them. The sponges keep dissolving. The parrotfish keep biting. The urchins keep rasping. And without new growth to replace what is being removed, the equation flips. The reef begins to lose material faster than it gains it. It is not a dramatic collapse. It is a slow, steady erosion. A net loss of limestone that manifests over years as a gradual flattening. A loss of structural height and complexity. A simplification of the three-dimensional architecture that every other organism on the reef depends on. What happened in the Caribbean illustrates this with devastating clarity. In 1983, a waterborne pathogen swept through populations of the long-spined sea urchin Diadema antillarum across the entire Caribbean basin. Within months, more than 97% of the population was dead. The die-off was one of the most catastrophic mass mortality events ever recorded for a marine invertebrate. Diadema had been the dominant herbivore on Caribbean reefs, keeping algal growth in check through relentless grazing. With the urchins gone, fleshy algae exploded across reef surfaces that had been kept clean for millennia. The algae smothered coral. It blocked light. It occupied settlement space that larval corals needed. Combined with already declining coral cover from disease and warming, the loss of this single herbivore tipped reef after reef from coral-dominated to algae-dominated states. Decades later, many Caribbean reefs have still not recovered. The urchin population has shown only partial signs of rebuilding, and the reefs that once rivaled the Pacific in structural complexity have been reduced to low, algae-covered rubble fields that bear little resemblance to what they once were. The lesson is not that bioerosion is the enemy. It is that bioerosion is essential, and that the organisms responsible for it are as critical to reef health as the corals themselves. Parrotfish grazing keeps algae in check, preventing it from overgrowing and suffocating coral. Boring sponges and worms create internal space that other organisms colonize. The rubble produced by bioerosion is raw material that, when cemented by crustose coralline algae, becomes new reef rock. The sand that settles in lagoons and channels creates habitats for seagrass, for juvenile fish, for burrowing invertebrates. Destruction in the reef's economy is not the opposite of construction. It is its partner. The reef needs both. And when you step back far enough to see the reef not as a single living community, but as a planetary scale chemical operation, the partnership between building and breaking takes on a significance that extends far beyond any individual reef system. Coral reefs are the largest source of biogenic calcium carbonate on the planet. The numbers are difficult to grasp. Current estimates place global reef carbonate production somewhere between 700 million and 2 billion tonnes per year. That is not a measurement of how much coral exists. It is a measurement of how much new limestone the world's reefs collectively manufacture in a single year, pulling calcium and carbonate ions from seawater and depositing them as solid mineral on the continental shelves of the tropics. The reef is not merely a biological community. It is a geological engine, a system that actively converts dissolved ocean chemistry into rock. The scale of this operation is constrained by three master variables, and their interplay defines where reefs can and cannot exist. The first is temperature. Reef-building corals, the species that harbour zooxanthellae and produce limestone fast enough to construct frameworks, are confined almost entirely to waters warmer than roughly 18 degrees Celsius. In practice, this restricts them to a belt stretching approximately 25 degrees north and south of the equator, a band that wraps around the Earth's midsection like a living geological belt. Step outside that thermal envelope and the corals survive but cannot build. They grow too slowly, their symbiotic algae function too inefficiently, and the energy budget does not close. The result is that reef construction is a tropical phenomenon, a consequence of physics and biochemistry that no amount of biological ingenuity can overcome. The second variable is the aragonite saturation state of the surrounding seawater. Aragonite, the specific crystal form of calcium carbonate that corals produce, can only precipitate readily when the water around the reef is supersaturated with respect to the calcium and carbonate ions that compose it. In warm, shallow tropical waters, this condition is met easily. The water contains more dissolved calcium and carbonate than is needed to maintain equilibrium, and the excess is what the coral harnesses to build its skeleton. As water deepens, cools and acidifies, the saturation state falls. Below a certain depth, the chemistry reverses entirely. Calcium carbonate begins to dissolve faster than it can form. This boundary is called the carbonate compensation depth, and in most of the world's oceans, it lies roughly 4 to 5 kilometres below the surface. Below that line, no limestone accumulates. Every shell, every skeleton, every fragment of carbonate that sinks to the deep ocean floor is dissolved back into its constituent ions before it can become rock. The reef exists in a chemical sweet spot, shallow, warm, supersaturated water where the physics favours precipitation over dissolution, and the organisms exploit that advantage with extraordinary efficiency. The third variable is light. The zooxanthellae that power the reef's energy budget are photosynthetic, and their productivity is directly proportional to the intensity of the light they receive. The most vigorous reef growth, the zone where calcification rates are highest and the carbonate factory operates at maximum output, is concentrated in the uppermost 10 metres of the water column. Below that, light attenuates rapidly. Corals still grow at 20, 30, even 60 metres depth, but their growth rates decline steeply, and the contribution to overall reef accretion diminishes accordingly. The reef's most productive construction zone is a narrow horizontal band near the ocean surface, a factory floor bathed in tropical sunlight. These three constraints, temperature, chemistry and light, together define the global geography of reef building. They explain why reefs are absent from cold waters, from deep waters, and from waters too turbid to let light through. They explain why the richest, most productive reefs on Earth are found in the shallow, clear, warm waters of the Indo-Pacific, and why reef development thins out and eventually ceases as you move toward the poles. They are not biological limitations, they are chemical and physical ones, and the biology has simply evolved to exploit the conditions in which the chemistry works. And here is something that cuts against every intuitive assumption about reefs and their place in the global environment. The act of building a coral reef, the chemical reaction that deposits calcium carbonate from seawater, does not remove carbon dioxide from the atmosphere. It releases it. When a coral precipitates one molecule of calcium carbonate, the reaction produces one molecule of carbon dioxide as a byproduct. The chemistry is unambiguous. For every unit of limestone the reef creates, a unit of carbon dioxide is generated. Reefs are not carbon sinks. They are, in strictly chemical terms, carbon sources. Think about that for a moment. The largest biogenic construction project on Earth, the one that has been running continuously for hundreds of millions of years, building structures visible from orbit, sustaining a quarter of all marine life, is simultaneously releasing carbon dioxide into the ocean and ultimately into the atmosphere. The reef builds its body from the same chemistry that warms the planet. The relationship between reef construction and global carbon cycling is real, measurable, and deeply counterintuitive. It does not diminish the ecological importance of reefs, which provide irreplaceable habitat, coastal protection, and economic value. But it does complicate any simple narrative that positions reef conservation as a direct tool for carbon management. The reef serves the living world in ways that have nothing to do with carbon accounting, and its value lies in what it builds, what it shelters, and what it sustains, not in what it removes from the atmosphere. What makes that value so difficult to measure and so easy to underestimate is the timescale on which it operates. Because the reef does not think in years, it does not think in decades. It thinks, if the word can be used at all for a system without a mind, in millennia. Try to hold a number in your mind. 0.5 centimetre per year. That is the growth rate of a typical massive coral, the kind that forms the structural bedrock of a reef. 0.5 centimetre. Not per day, not per month, per year. In the time it takes a human child to grow from infancy to adulthood, a massive coral colony has added roughly 9 centimetres to its skeleton. In the time it takes a civilization to rise, flourish, and decline, that same colony might have grown a few metres. The reef does not operate on any time scale that the human mind finds natural. It operates on geological time, and the gap between our intuition and its reality is one of the hardest things about understanding what a coral reef actually is. The living Great Barrier Reef, the one visible from satellites, the one that tourists visit and scientists study and governments argue about, established itself on the flooded continental shelf roughly 6,000 to 8,000 years ago. Think about what that means. The reef predates the Egyptian pyramids by thousands of years. It predates Stonehenge. It predates the invention of writing, the first cities, the first agriculture. When human beings were just beginning to domesticate wheat and barley in the Fertile Crescent, the corals of the Queensland coast were already decades into their current building project. When the first pharaoh unified Upper and Lower Egypt, the reef had been growing continuously for more than 3,000 years. It is one of the oldest living construction projects on the planet, and it has never stopped, not for a single year, not for a single day. And the reef you see today is only the most recent surface of something much older. Beneath the living coral, beneath the first few metres of recently deposited skeleton, lies limestone that was laid down by corals that died centuries ago, millennia ago, tens of thousands of years ago. The platform beneath the Great Barrier Reef's modern surface extends in places to more than 100 metres of depth. And the oldest material at the base of those formations dates back hundreds of thousands of years. The living reef is a veneer on a monument. The monument was built by the ancestors of the organisms building the veneer, and their ancestors before them, and theirs before them, in an unbroken chain of construction that stretches back through ice ages, through sea level catastrophes, through mass death events from which the reef recovered and kept building. This is what makes coral reefs unlike almost anything else in the living world. Most biological structures are built by a single generation and abandoned when that generation dies. A bird's nest lasts a season. A beaver dam lasts a few years. Even the most impressive termite mounds are rebuilt from scratch when the colony expires. But a coral reef is inherited. Each generation of polyps builds directly on top of the skeletons left behind by every generation before it. The young colony does not start from nothing. It starts from the accumulated physical legacy of millennia of prior construction. A limestone platform that was there before it arrived and will be there long after it dies. The coral does not just inherit genes, it inherits architecture, it inherits geology, it inherits a place in the world that was built, grain by grain, by organisms that are long gone, but whose contribution remains permanently underfoot. The patience required to build a structure like the Great Barrier Reef is almost impossible to convey in human terms, but the arithmetic is simple enough. 0.5 centimetre per year, sustained across 8,000 years of continuous growth, produces roughly 40 metres of vertical reef. That is the scale of what has been accomplished since the modern reef began. 40 metres of solid limestone, manufactured from seawater by soft-bodied animals smaller than a grain of rice, with no plan, no coordination, no awareness that anything was being built at all. Each polyp simply did what polyps do. It fed. It photosynthesized through its algae. It precipitated a thin layer of aragonite beneath its body. It budded. It died. And the next generation did the same, on top of its skeleton, in the same spot, using the same chemistry. The magnificence of the reef is not the product of intent. It is the product of repetition. The same small act, performed by billions of organisms across thousands of years, without variation, without pause, without any concept of what the cumulative result would be. There is one group of people for whom the reef's timescale is not abstract. Along the Queensland coast, over 70 Aboriginal and Torres Strait Islander groups hold cultural connections to the waters and reef systems that Western science calls the Great Barrier Reef. Their relationship with this place is not measured in the centuries since European arrival. It is measured in tens of thousands of years. Their ancestors fished these waters, navigated these reefs, and developed intimate knowledge of their rhythms long before the modern living reef existed in its current form. When sea levels were lower during the last ice age, and the continental shelf was dry land, the people who would become the traditional custodians of this coast walked where the reef now grows. Their oral traditions carry knowledge of landscape changes, of coastlines shifting, of islands appearing and disappearing that aligns with what the geological record confirms. They are the only continuous human witnesses to the reef's deep history, and their concept of sea country, a framework that understands the reef not as a resource to be managed, but as a living system to which people belong, offers a perspective on timescale that Western science has only recently begun to appreciate. The reef builds in silence. It builds without urgency. It builds with a consistency that makes human persistence look frantic. And it has been doing this, in one form or another, for so long that the question is not whether it will continue, but whether the conditions that allowed it to continue for hundreds of millions of years are now changing faster than it can adapt. They are. The ocean is warming. This is not a projection, it is a measurement. Sea surface temperatures in the tropics have risen by roughly 0.7 degrees Celsius over the past century, and the rate of increase is accelerating. 0.7 degrees sounds trivial. It is not. Coral bleaching, the breakdown of the symbiosis that powers the entire reef, is triggered when water temperatures exceed the local summer maximum by just 1 to 2 degrees. The margin between health and catastrophe is vanishingly small, and the warming trend is pushing reef systems across that margin with increasing frequency. The record speaks for itself. the Great Barrier Reef experienced severe mass bleaching in 2016, again in 2017, again in 2020, again in 2022, again in 2024. 5 mass bleaching events in 9 years. The intervals between them are shrinking. Research published by a team working out of James Cook University documented that the time between severe bleaching events on reefs worldwide has halved since the 1980s. Reefs that once had a decade or more to recover between thermal stress events are now being hit again before the recovery is complete. Each successive bleaching kills a proportion of the coral. Each recovery is partial. The trajectory is clear, and it is not a trajectory towards stability. Warming is not the only chemical assault. The ocean is also acidifying. As carbon dioxide concentrations in the atmosphere rise, the ocean absorbs a significant fraction of that gas, and when carbon dioxide dissolves in seawater, it forms carbonic acid, which lowers the pH of the water and reduces the concentration of carbonate ions available for corals to build their skeletons. The chemistry is straightforward and its consequences are measurable. Research conducted at the Woods Hole Oceanographic Institution showed that under business as usual emission scenarios, ocean acidification could reduce coral skeletal density by up to 20%. The corals would still grow. They would still deposit aragonite, but the skeleton they produced would be thinner, weaker, more porous, less able to withstand the physical forces, the storms, the wave energy, the bioerosion that a reef must endure to persist. The buildings would still go up, but the walls would be made of something closer to chalk than to stone. And then there is sediment. The geological record shows with painful clarity that the Great Barrier Reef has always been most vulnerable to smothering. Of the 5 death events documented in the 30,000-year drilling record, the one that was most difficult for the reef to recover from was not caused by temperature change or sea level shift. It was caused by an influx of terrestrial sediment, mud and silt washing from the land and burying the reef beneath a layer of material that blocked light, clogged polyps, and suffocated the living surface. That vulnerability has not changed. What has changed is the volume of sediment reaching the reef. Agricultural runoff, land clearing, and coastal development along the Queensland coast have dramatically increased the sediment load carried by rivers draining into reef waters. The threat that brought the reef closest to permanent collapse 10,000 years ago is now being amplified by human land use. Each of these stresses, warming, acidification, sedimentation, is something the reef has faced before in its geological past. The drilling record proves it. The reef died 5 times in 30,000 years and came back each time, migrating across the seafloor, recolonizing new substrate, rebuilding with the same chemistry and the same patience that had always carried it through. But the past death events occurred one at a time, driven by a single dominant stressor, and they unfolded over timescales of centuries to millennia. The reef had time to respond. It had time to migrate. It had time to adapt. What is different now is the combination. Warming and acidification and sedimentation are arriving simultaneously, driven by the same underlying cause, and they are arriving at a pace that has no precedent in the reef's geological biography. The rate of temperature change alone is orders of magnitude faster than anything recorded in the coral cores. The reef's proven resilience was built for a world that changed slowly. Whether that resilience extends to a world that is changing this fast is the question that no amount of deep time drilling can answer. And there are threats that leave no trace in the fossil record at all. The breakdown of spawning synchrony documented in the Red Sea, where individual colonies of the same species have drifted weeks or months apart in their reproductive timing, is invisible in limestone. The interference of artificial coastal lighting with the moonlight cues that corals depend on to coordinate their spawning is a phenomenon that did not exist before the 20th century and has no geological analogue. These are novel pressures, stresses that the reef's evolutionary toolkit was never designed to handle, and their effects are subtle, cumulative, and very difficult to reverse. There are people trying. At research facilities in Australia, teams of marine biologists are working on what amounts to selective breeding for corals, crossing strains from warmer northern reefs with strains from cooler southern reefs to produce offspring that may tolerate higher temperatures. The program is called assisted gene flow, and it represents a deliberate human intervention in a process that has been running on its own for hundreds of millions of years. The idea is not to replace natural selection, but to accelerate it, to give the reef's own adaptive machinery a head start against changes that are arriving faster than evolution can normally respond. Whether it will work at scale across a reef system that spans 2,600 kilometres remains an open and genuinely uncertain question. There is also a quieter concern, one that does not make headlines. It is called dark diversity, and it refers to the species that should be present on a reef based on its environmental conditions and geographic location, but are not. Their absence is a signal. It suggests that past disturbances, events that may have occurred years or decades earlier, have left a legacy of loss that the reef has not yet recovered from. A kind of ecological debt that accumulates silently and may not become visible until it is too late to repay. A reef can look functional. It can have coral cover. It can support fish. And it can still be missing species that were once there. Species whose absence weakens the system in ways that may only become apparent when the next stress arrives and the expected resilience does not materialise. The reef has survived everything the planet has thrown at it for 30,000 years. It has survived ice ages, drowning, exposure, and burial. It has migrated, rebuilt, recolonised, and persisted with a tenacity that makes it one of the most resilient biological systems ever documented. The question is no longer whether the reef can recover from catastrophe. It has answered that question 5 times. The question is whether it can recover from this one. Go back to the beginning. Go back to where this story started. A single polyp, soft-bodied, blind, brainless, no larger than a grain of rice, sitting on a hard surface somewhere in the warm shallows of a tropical ocean. It has no plan. It has no awareness of what it is about to do. It simply does what its chemistry dictates. It pulls calcium ions from the water around it. It pulls carbonate ions. It concentrates them in the thin fluid space beneath its body, raises the pH, and precipitates a crystal of aragonite. One crystal, then another, then another. A thin disc of mineral forms beneath its tissue, the first foundation stone of a skeleton it will never finish building. That polyp buds. The clone buds again. A colony emerges, genetically identical, connected by living tissue, each polyp adding its own thin layer of limestone to the growing surface. The colony expands outward and upward, its shape dictated by the interplay of light, current, gravity, and the specific genetic instructions encoded in every cell. Branching or massive, plating or encrusting, the form varies, but the act is the same. Calcium carbonate, extracted from seawater, deposited as solid mineral, accumulated grain by grain across years, across decades, across centuries. When the polyps die, their skeletons remain. The next generation settles on top. The process continues. It has been continuing in one form or another for 240 million years. From ion to crystal. From crystal to skeleton. from skeleton to colony, from colony to reef, from reef to a structure so vast it is visible from orbit, stretching 2,600 kilometres along a continental coast, covering an area larger than the United Kingdom, supporting a quarter of all known marine species in an ecosystem built entirely from the accumulated output of animals that weigh almost nothing and live for a handful of years at most. The arithmetic is absurd, the patience is incomprehensible, and the result is one of the most extraordinary things that life has ever produced on this planet. You have heard the full arc of this story now. You know what powers the construction, the symbiotic algae that turn sunlight into the energy a polyp needs to lay down stone. You know what controls the chemistry, the biological machinery that manufactures amorphous nanoparticles and guides their crystallization into aragonite with a precision that no human engineering has yet matched. You know how the reef reproduces, how moonlight synchronizes the spawning of more than 100 species on a single night, how larvae drift on ocean currents and settle on surfaces marked by chemical signals older than the dinosaurs. You know how the reef has survived. 5 catastrophic death events in 30,000 years, each followed by migration, recolonization, and rebuilding on a scale that redefines what biological resilience means. And you know what threatens it now? A convergence of warming, acidification, sedimentation, and reproductive disruption arriving simultaneously and at a pace that has no precedent in the geological record. The honest answer to the question of whether the reef will survive is that no one knows. Not the researchers drilling cores from the continental shelf. Not the marine biologists breeding heat-tolerant coral strains in laboratory tanks. Not the ecologists documenting the accelerating frequency of bleaching events. The reef has proven repeatedly and spectacularly that it can come back from the dead. But it has never faced what it faces now, and the tools it used to survive in the past, migration across the seafloor, vertical growth to track rising water, centuries of uninterrupted recovery time may not be available to it under current conditions. The reef may rebuild, it may adapt, it may find new configurations, new species compositions, new ways of persisting in a warmer, more acidic ocean. Or it may cross a threshold from which the return is not to what it was, but to something fundamentally different. A simplified, algae-dominated system that still exists, but no longer functions as the biodiversity engine it once was. Not extinction in the absolute sense. Transformation. A reef that is still there, but is no longer a reef in any way that matters. That distinction between functional extinction and ecological transformation may be the most important idea in modern reef science. It acknowledges that the question is not simply whether coral will survive. Coral will almost certainly survive in some form, somewhere, for a very long time. The question is whether what survives will still be capable of building the structures, supporting the communities, and sustaining the processes that make coral reefs the most biologically productive habitats in the ocean. A reef with 5% coral cover and 95% algae is technically still a reef. It is not the same thing. But here is what stays with you, long after the chemistry and the biology and the geology and the politics have faded. Here is the fact that is hardest to let go of. The reef does not know it is building anything. No polyp has ever conceived of a reef. No colony has ever intended to produce a structure visible from space. No generation of corals has ever understood that it was contributing to a construction project that would span millennia and reshape the geology of an entire continental shelf. Each organism simply does what it does. It feeds. It photosynthesizes. It precipitates a thin layer of mineral beneath its body. It reproduces, it dies, and the next one does the same on top of its skeleton, in the same water, under the same sun. The magnificent is not designed, it is accumulated. It is the consequence of patience multiplied by time, of a simple act repeated so many times, by so many organisms, across so many years, that the result transcends anything the individual builders could have imagined, if they could imagine anything at all. You are a temporary visitor to a construction site that was ancient before your species existed. The limestone beneath the living coral was being laid down when woolly mammoths walked the earth. The chemistry that drives the process was old when the first fish evolved jaws. The lineage of builders stretches back to an era when the continents were arranged in a configuration you would not recognise on a map. And the reef is still building, right now, tonight, this year, adding its 0.5 centimetre, its thin layer of aragonite, its modest contribution to a project that has been running for longer than the human mind can meaningfully comprehend. Whether it will still be building a century from now depends on decisions being made by a species that arrived on this planet roughly 300,000 years ago, and has been aware of coral reefs for less than 300 years. The reef has been here for millions. It does not need us to build. It needs us to let it. The polyp does not know what it is making. It never has. It simply builds, and the ocean does the rest.